Welcome back, friends. Welcome back in another video tutorial for microbiology with Shagni. So welcome back in another video tutorial from the playlist of mechanism of action of various type of enzyme. So today we will going to do the last video in this playlist, that is the mechanism of action of lysozyme. We have started our video lecture series with chymotrypsin. Then we have talked about aspartate protease. Then in just previous one we have talked about RNase A, and last today's video we will going to talk about lysozyme. This this type of enzymes are very important and to break mainly the beta 1,4 glycosidic linkage. So let's get started with the video, friends. So lysozyme is a kind of enzyme which is mainly present in secretions such as tears, sweats, and not only secretions but also it is present in egg white. We will going to use lysozymes which was isolated from the hen egg. So this enzyme was first described by Alexander Fleming, although the structure of the lysozyme was discovered by David Chilton Phillips in the year 1965. So it have the ability to break the beta 1,4 glycosidic bond that usually connects N-acetyl glucosamine and the fourth carbon of the it will be NAM and the fourth carbon of the NAG, right? That is the composition of the cell wall peptidoglycan, right? So how it will do is, first of all, this lysozyme belongs to the group of the enzyme called hydrolase. Hydrolase means it will facilitate hydrolysis, right? That means it will facilitate breaking of the beta 1,4 glycosidic bond by the addition of water, right? So that is the thing. So how it will do this kind of thing? This kind of thing it will do by binding to the peptidoglycan binding site. This is very important, and we have talked about this binding site when we have talked about the mechanism of action of penicillin also, right? So this kind of reaction is hydrolysis reaction which is catalyzed by this enzyme, as I have told you. And here a molecule of water will be added to the single bond between the adjacent sugar in the polysaccharide chain, thereby causing the breaking of the bond. We will going to see how it will occur, and it is obviously energetically favorable. So that is the idea. Now let's move to the mechanism of action. So in the mechanism of action of lysozyme, two amino acid will going to play very important role. First of all, glutamic acid, which is present at the 35th position. Another one is aspartic acid, which is present at the 52 position. This is this one. Okay. So now this is NAM. We have discussed about NAM in our cell biology lecture series, and this is NAG, right? And they are joined by beta 1,4 glycosidic bond, right? So now what will happen, that remember one thing, that in the mechanism of action of lysozyme, this glutamic acid is uncharged, but this aspartic acid is charged. Okay, and it is probably due to the change in pH in their local environment. Okay, so now what will happen, that it will donate this glutamic acid will going to donate a proton, right? And it will taken up by this NAG, and that's how it will get the OH. Okay, and now the carbonium ion which is generated here will be stabilized by this aspartic acid. Okay, there will be a formation of bond here. So what we will get? We will get something like this. Okay, now a water molecule will come into the picture, and the water molecule will donate hydroxyl to this, and the remaining proton will be taken up by the glutamic acid, and as a result the glutamic acid, which becomes charged due to the donation of the proton, becomes again uncharged. That means the glutamic acid comes back to its normal state, right? And now due to this reaction, OH will come here, H will come here, and aspartic 52 also released, and now it will become again charged. Okay, so you can see it in the last part of this reaction. Okay, so that is the idea of how the mechanism of action is going on in case of lysozyme. So for a quick recap, two amino acid in the enzyme will going to play very important role. Glutamic acid 35, aspartic acid 52. Glutamic acid here is uncharged, aspartic acid is charged, right? Glutamic acid will do what? Glutamic acid will going to donate the proton. And aspartic acid is charged and it will going to stabilize the carbonium ion generated due to the donation of glutamic acid 35. Now, in the next part of the video, water will come into the picture and it will donate its hydroxyl to this NAM and it will donate the proton to this NAC, right? And as a result, the aspartic 52, which was involved in stabilizing this ion, will be lived or will be released, right? And the proton will be taken up by the glutamic 35. So that's how the beta 1,4 glycosidic bond between two lysozyme will be cleaved. Okay, so I hope this video will be helpful for you for understanding the concept of lysozyme and its mechanism of action. So thank you for listening to this class. We will continue our enzyme lecture series and we will do now about the allosteric enzyme. Okay, so till then, bye bye.